So we've got two separate energy structures. Think of it as fat being one, which can be stored or used for energy, glucose being another one, glucose being used or stored as energy. Now, here's the key thing to understand, and this is what the Randle cycle is all about. And what they studied is because all they had were these substrates. They had insulin. They knew about insulin from the diabetes days, from Banting and Best's work in the 1910s, and they knew about fat, and they knew about glycerol, and they knew about sugar, glucose. They didn't know anything else. And that's very important. In 1963, they did not have knowledge of anything else. So when they studied these muscle cells in culture, so they grew the muscle cells in a petri dish, and they influenced them by various things. So one of the things they did is they didn't provide insulin, or they created diabetes, type 1 diabetes, by using in animals, a particular toxin that killed the insulin-producing cells. Okay? And what they did then is they also used growth hormone, they used cortisol, they used a variety of different hormones that they didn't know about to see what influenced these cells. And what they found is the following. They found, number one, that muscle cells preferentially prefer to metabolize non-esterified fatty acids. So when you have adequate supply of fatty acids or of ketones or of fat to the cell, and it's an otherwise healthy cell, irrespective of insulin, the preferential pathway is to use that fat as the primary source of energy for the muscle. The fat preferentially goes into the mitochondria and becomes ATP. And if anybody out there is an athlete, that is such a crucially important thing to understand, is that the muscle cells preferentially use fat, and fat is mostly available between meals. And if you are going long periods of time, like we did in the 1960s, where we didn't snack all day long, we were going long periods of time eating 2, maybe 3 meals a day, those long periods of time, fat was dominant in our bloodstream. The fat cells were releasing fat into the bloodstream. Fat was floating in the bloodstream, either attached to albumin or being transported by LDL that they did not know about. But the fat came to the cells, and the liver was producing ketones, and muscle cells, heart muscle cells, and striated muscle cells preferentially use non-esterified fatty acids as their source of energy. In fact, there's another paper, which was subsequently published in 1985, I believe, that looked at the different types of energy used by the heart muscle. And we know that the heart muscle, everybody's heart muscle, about 85% of the energy to the heart muscle is non-esterified fatty acids.